Okay, hello everybody. Just a few tests for sound and video. Do you hear me?
Yep.
Okay, so everything seems to work perfectly. We can start. So hello everybody, I am Alberto LERNER, the president of CHROMagar. Thank you for attending this webinar where we will talk about a very hot topic, Candida auris, and what we have done at CHROMagar about it, the new CHROMagar Candida Plus. I would like to thank particularly our speakers of today, not only for sharing their experience on this topic with us, but also because they carried the evaluation of this new formulation during the peak of the pandemic, which was certainly a very tough work. So the agenda for today. First, I will make a presentation of what we do at CHROMagar. Then we will listen to the presentation of Dr. Nuria TORMO-PALOP, then the one of Dr. Carme Salvador-GARCIA, and at the end, the one from Professor Andrew BORMAN. At the end, we will have some minutes to answer to your questions. So please attendees, use the Q&A system of the Zoom at the bottom of your screen to send us your questions. If we don't have time to reply to all the questions, no worries, we will send you an email with the replies. Now we would like to ask you, to everybody, to send us a message with the chat system with the country where you are. And also send us a guess, the number of countries you think will be represented today by the attendees.
Sweden, Colombia.
And hopefully by the end of the webinar, we will be able to count the number of countries and we will tell you. The persons who guess the correct number will get one of the most delicious French chocolates. Okay?
Okay, let's start. CHROMagar is a French company. Our laboratory is located in Paris, in the city center, a few minutes walk from the Eiffel Tower. You are all invited. When you come to Paris, please come to visit our laboratory. All what we do, and we do it very well, is chromogenic culture media. All our products are available in dehydrated form, and in most of the countries, they are also available as ready-to-use Petri dishes, freshly made by local authorized manufacturers. A little bit about the story of the chromogenic technology pioneered by Dr. Alan Rambach, the person you see in this picture. When he was doing his PhD in the Pasteur Institute in the late 70s, he had the idea to make the microorganism grow in different colony colors to make easier the reading of the plate. When he managed to color in black the Escherichia coli, the bacteria he was working with, he understood that this technology should be interesting not only for research laboratories, but also for routine microbiology. So he filed a patent in 1979. And today, more than 40 years later, the idea he envisioned proved to be correct. Since the first patent in 1979, we continue to develop new formulations, and we try to launch 1 or 2 new formulations per year. We have already about 40 different media. Today, we will talk about one of the most recent ones, CHROMagar Candida Plus. The idea was to modify our legendary formulation so to get a very specific aspect for Candida auris. Why were we interested in improving Candida auris specificity? We shall explain it in the coming slides. What's the difference between traditional media and CHROMagar media? When I say traditional media, I mean agars like maconche, XLD, mannitol salt. In all these agars, you detect the presence of bacteria when they digest a sugar, which will cause a change in the acidity of the agar and a change of the color of the pH indicator. In this example, a maconche plate, the lactose fermenters will turn red and the non-fermenters will remain unchanged. In CHROMagar media, we do not use anymore the combination of sugar and pH indicator. We target very specific enzymes with wisely chosen substrates. The enzyme cleaves the chromogen and releases a precipitate, which will color the colony of the microorganism we want to detect. So, as you can understand, with this technology we can achieve better specific differentiation, particularly interesting in samples with mixed complex flora. Okay, that's for our technology. Now let's talk about the importance of Candida auris. As you probably have seen, in the recent years there have been several alerts about Candida auris. Here are some examples. In 2015, the American CDC. In 2017, the Pan American branch of the WHO. In 2018, the European CDC. The first detailed study on Candida auris was published by Professor Makimura and his team in Japan in 2009. And since then, there have been reports worldwide with increasing frequency, spreading very rapidly, as shown in this recent paper from CHROMagar and colleagues. Professor BORMAN will talk later on more in detail about this. But in fact, Candida auris seems to be spreading faster than we can publish. For instance, we know that there was already a first report of Candida auris in Brazil. Here I have selected a few reports showing the increasing prevalence of Candida auris in the European countries, where not only the number of bloodstream infections has grown, but also the ratio of colonizations. In the US, the increasing number of clinical cases in the last 5 years. And a very fresh and very worrying diagram from an alert of the California Public Health Department showing the correlation between the number of COVID-19 cases and Candida auris cases in the last few months. When you search for Candida auris publications in PubMed using the words Candida and auris, you can see that there have been more than 1,000 publications and very concentrated in the last 5 years with an exponential growth. Here are some examples of these really worldwide publications. I would like to draw your attention to this literature review from Jeffrey Smith and colleagues, compiling evidence of misidentification of Candida auris by biochemical-based systems, but also by Malditov. So because of all this, we decided to modify our CHROMagar Candida to render much more specific the aspect of Candida auris. As you can see in the picture, even for a very experienced mycologist, in the Saburo agar, you will not be able to differentiate much. In our new medium, we managed to maintain the good differentiation between the most common Candida species, Candida albicans in green, Candida tropicalis in metallic blue, Candida krusei fuzzy pink, Candida glabrata in mauve, And the new aspect of Candida auris, bluish colonies surrounded by a halo, and blue from the back of the plate. Let me show you some variations of this yeast aspect on different isolates. All these are Candida auris strains from different clades. As you can see, the intensity of the coloration may vary from some darker, some lighter, but still it is immediately recognizable. This new medium was evaluated in many countries on many isolates from different collections. More than 500 isolates from various Candida species, including more than 200 Candida auris. And all these with astonishing results in terms of sensitivity and specificity. Here you see some examples. Following these worldwide evaluations, several organizations are already citing our new medium as a good tool for epidemiological surveillance cultures targeting Candida auris. Here an example of the February 2021 epidemiological alert of the Pan American branch of the WHO. By the way, you can see in this report that the first cases in Brazil and Peru have been already added. I showed you a few minutes ago a graph from California presenting a correlation in the number of cases of Candida auris and COVID-19. The same situation has been reported in several other countries in healthcare units dedicated to COVID-19 patients. In Mexico, in India, in Florida in the US, in Lebanon, and also in Colombia. Also, I would like to remind you that Candida auris has shown astonishing capacity to survive on inanimate surfaces for days. In case of outbreaks, you may want to screen not only patients at risk, but also their environment. Well, that's about all what I wanted to tell you on our CHROMagar Candida Plus. Before I pass the microphone to our guest, I would like to remind you that at CHROMagar we have developed many other chromogenic media for clinical specimens. Some of them designed for clinical routine, like CHROMagar orientation for urinalysis, CHROMagar strep A, strep B, CHROMagar staph aureus, and also for screening antibiotic resistant bacteria. The first one launched more than 20 years ago, CHROMagar MRSA, and several other topics like VRE, ESBL, carbapenem resistance, and the latest one, CHROMagar Lean-R, to screen for linezolid resistant gram positives.
Okay, so thank you very much for listening to my presentation. I hope you now have a better idea of what we do at CHROMagar. If you are interested in collaborating with us, scientific or commercial, please do not hesitate to contact us. Our email address is chromagar@chromagar.com. I remind you, you can send questions to our speakers through the question and answers of the Zoom system at the bottom of your screen. Okay, now we will listen to the presentation of Dr. Nuria TORMO-PALOP, lessons learned from a 3-year outbreak of Candida auris in a tertiary hospital.
Good morning. My name is Nuria TORMO-PALOP, and I work as a consultant microbiologist in the Consorcio Hospital General Universitario of Valencia, in Spain. I am going to present a 3-year outbreak of Candida auris in my institution. Candida auris is an emerging fungal pathogen that presents a serious global threat. It is concerning for 4 main reasons. First of all, its rapid spread. This species has spread worldwide in a decade, since its first description, where it was isolated from the auditory canal of a Japanese patient in 2009, until now, 2021. That transmission in multiple cases have been reported in more than 30 countries and 6 continents, as you can see here in the CDC map. In the right, you can see a timeline showing the expanding worldwide detection of Candida auris. Candida auris can spread in healthcare settings because it has the ability to survive in the environment, in plastic biomedical devices such as catheters, surfaces, and equipment, with persistence on patient skin for a long period of time, with a high rate of patient-to-patient transmission, thus showing the importance of colonization and hand hygiene. As you can see in that picture, it has been isolated from many surveillance samples, being the most frequent the axilla and the groin. In this other picture, we can see that the number of Candida auris cases declared in Europe, in gray, are mainly from carriage. These characteristics explain that Candida auris is able to cause nosocomial outbreaks, which have been reported in several countries, especially in intensive care units. Here, we can see the situation in Europe, with declared outbreaks in red in Spain and in the UK. Candida auris, secondly, can cause invasive infections. Those infections have high mortality, 59% all-cause mortality in early studies, although in more recent studies it is not so clear. A huge number of the cases are bloodstream infections, candidemia, as shown in the annual report of cases in Europe, in blue, where candidemia is the most frequent declared infection. You can see it there. But Candida auris has been isolated from the respiratory tract, the brain, abdominal cavity, urine tract, bones, etc. Third, Candida auris is often multidrug resistant, so that the treatment is often more difficult. 4 geographic clades, well, now there is a new one, have been described, and there are differences in resistance among those clades. Apart from the East Asian clade, or clade 2, found mainly in Japan and South Korea, which is susceptible to all antifungals in a high proportion, all the other clades have high-level resistance to fluconazole. Moreover, the South Asian clade, or clade 1, in dark gray here in the map, has the highest resistance to fluconazole and amphotericin B. And there are strains belonging to that clade which have resistance to the 3 major classes of antifungals. Here in Spain, the studied isolates belong to the South African clade, or clade 3, while those in the rest of Europe are clade 1. The fourth characteristic of Candida auris is that it is difficult to identify. Most often it is misidentified as Candida emuloni by conventional biochemical methods. Mass spectrometry or DNA sequencing are required. Misidentification may lead to inappropriate management. Now I'm going to present how the Candida auris outbreak took place in our hospital, which is a 500-bed tertiary hospital. In September 2017, a patient was admitted from another hospital of our city in which there already existed a declared Candida auris outbreak, La Fe. This patient had a known Candida auris urinary tract infection and was not admitted to the ICU in our hospital. A month later, in October, we had the first case of candidemia in a patient who was hospitalized in the ICU and had not been hospitalized in other centers. Promptly, we performed a screening colonization of all the patients of the ICU, which now accounts for 40 beds, and 4 patients were positive. As we already had the protocol of obtaining surveillance samples for multidrug resistance gram-negative microorganisms, it was quite easy to add to the same samples a yeast culture, in order to screen for Candida auris carriage. Briefly, all the patients are screened at ICU admission and weekly. From there to the present date, we have 500 colonized or infected patients with 162 positive invasive clinical samples, among them 80 candidemias and 1 meningitis. According to antifungal resistance, we have high resistance to fluconazole in almost all the isolates, while resistance to amphotericin B and echinocandins is scarce. Here is depicted the evolution of species distribution causing candidemia since 2011 in our hospital. As you can see, Candida albicans in red was the most isolated species until the appearance of Candida auris in 2017, followed by Candida glabrata in gray and Candida parapsilosis in yellow. In 2018, we had a large increase in the total candidemia episodes, with 65 cases, mainly due to the high number of Candida auris cases, 21, as the number of the other species cases was almost comparable to the previous years. Then in 2019, we had a decrease, probably because of the good implementation of infection control measures to stop spreading of Candida auris. But in 2020, last year, with the COVID-19 pandemic, we had the highest number of candidemia cases, more than 70, also due to the increase of Candida auris cases, which were 33 in total, while Candida albicans accounted for 15 cases, like the years before. Also, in the first 3 months of 2021, we have had also a high number of cases, as you can see here. In the following figure, we can see the follow-up of the Candida auris candidemia and colonization cases from the beginning of the outbreak. We have added the incidence of SARS-CoV-2. Since Candida auris appeared in our setting, we started a surveillance study of patients at admission and weekly in the ICU, where the outbreak was set. By the end of 2018, after applying strict prevention and control measures, the number of Candida auris colonization and invasive infections decreased drastically. Here you can see 2019 with a few cases. Then, with the COVID-19 pandemic, we suffered the consequences of the healthcare system pressure due to COVID-19 admissions at the hospital, leading to reorganization of the wards and relaxing the control measures. The worst numbers are found at the beginning of the present year, 2021, taking into account that the worst wave of COVID-19 in Valencia, our city, has been the third one. So we can see how the number of cases of Candida auris colonization and then invasive infections has increased in the last year, being the highest ever since it started. In July 2020, we published the following article in which we described the characteristics and management of Candida auris candidemia episodes. In this table, we can see the epidemiological and clinical characteristics, antifungal treatment, and outcome of the episodes we had at that date. It is important to note that the median length of hospital stay before the candidemia episode was 29 days, being 91.5% hospitalized in the ICU or discharged from there to a general ward. Almost 75% of the patients with candidemia had a previous positive screening culture. According to patients' characteristics, age, Apache 2, and Charlson comorbidity scores were relatively low. The most frequent underlying disease of the patients with Candida auris candidemia was gastrointestinal disease. First abdominal surgery, followed by not surgical, which is equally common with other Candida species. Among the most prevalent risk factors in our series, we found previous antibiotic treatment, the use of central venous and urinary catheters, mechanical ventilation, and previous surgery, previous colonization, and prolonged stay in the ICU, over 2 weeks. Almost all of the patients were treated with echinocandins, in monotherapy, half of the patients, or in combination with amphotericin B or isavuconazole. In 70% of the cases, the catheter was also removed. We had accrued 30-day mortality of 23.4%, which is less than previous reports that estimated 30 to 60% of mortality. And interestingly, we found 2 cases of endophthalmitis, which is more common in other Candida species, as Candida albicans. To conclude, we can say that we have a more than 3 years outbreak of Candida auris, which we may not call an outbreak anymore, but an endemic situation. We found a high number of colonized patients because of healthcare spread, despite all the infection control measures applied, mainly in the intensive care unit. The number of candidemia episodes in the hospital has increased due to the appearance of Candida auris. 75% of the Candida auris candidemia episodes are preceded by positive screening cultures, thus raising the importance of such cultures. Fortunately, the resistance to antifungals remains more or less stable over the years. Anyway, the consumption of antifungals has increased in the last period, probably due to the increase of candidemia. What we can do to stop or at least minimize the spread is to follow strict infection control practices, including surveillance studies. And that's all. Thank you for your attendance.
Okay. Thank you very much, Nuria, for this interesting story about the Candida auris outbreak in your hospital. I can only imagine the additional stress that might have caused in addition to the COVID-19 patients management. Okay, before we go to the third presentation, I would like to remind you that you can use the question and answers system of the Zoom at the bottom of your screen to send us your questions. Okay, so now we will watch the presentation of Dr. Carme Salvador-GARCIA, CHROMagar Candida Plus experience in a tertiary hospital.
Hello everyone, good afternoon. My name is Carme Salvador-GARCIA. I'm a clinical microbiologist from the General University Hospital of Valencia in Spain. First of all, I want to say thank you to CHROMagar for the invitation to this webinar. Today, I'm going to explain our experience with CHROMagar Candida Plus plates. Before going to any further, I would like to introduce briefly the center where I work. The hospital is located in Valencia, a city in eastern Spain. The history of the hospital dates back to February in 1409, when Friar Joffre committed to set up the need to raise a hospital for poor people that until then lacked of any assistance. Currently, the hospital has a population of 350,000 and has more than 500 beds. As my colleague, the doctor Nuria Tormo said, in September 2017, Candida auris was detected for the first time in our hospital. In the figure, you can see the temporal evolution of Candida auris colonizing patients, which is the green line, and candidemia cases, the purple one. In May 2020, we conducted a study with CHROMagar Candida Plus plates in our hospital. In August, we published the results that I would like to comment in the following slides. The aim of the study was, first, to compare the performance in the novel chromogenic medium CHROMagar Candida Plus with CHROMagar Candida for the presumptive colorimetric identification of Candida species. And second, to evaluate the utility of the novel chromogenic medium CHROMagar Candida Plus in the detection of Candida auris in surveillance samples. How can we possibly conduct the accurate steps to research these objectives? In order to achieve the first objective, we prepare a total of 39 suspensions containing different mixtures of strains of Candida. The table shows the different Candida species specimens. And to achieve the second objective, a total of 25 swabs from surveillance studies, axial, rectal, and pharyngeal samples submitted to the microbiology department were selected. Suspensions and swabs were inoculated in both plates. Plates were then incubated at 37 degrees Celsius and read at 24, 36, and 48 hours independently by 2 qualified laboratory staff. We analyzed the color and size of the colony. Finally, the colonies were presumptively identified by colony color, following the manufacturer's instructions in both medium, and the identification was always confirmed using MALDI. What results were obtained? A total of 95 isolates were obtained by culturing the colonization swabs under strain suspensions. I'm going to start by describing the change in colony size at the different times. Examination of media at 24 hours showed that there wasn't sufficient time to differentiate correctly among species, given the small size of colonies. However, as you can see in the picture, Candida lusitania and Candida auris seem to grow better in the CHROMagar Candida Plus. At 36 hours, surprisingly, all Candida colonies generally became larger in CHROMagar Candida Plus. The greatest difference in size was found for Candida auris, Candida lusitania, and Candida crasei, as you can observe in the following images. In the same way, you can see in the photo that the difference in size in the colonies were more significant at 48 hours of incubation. Now, I'm going to evaluate the colony color. At 24 hours, most of colonies were colorless. There was not sufficient time to differentiate correctly among species. At 36 hours, color differentiation was possible for most species in the CHROMagar Candida Plus. Although some species had a similar bluish color, such as Candida auris and Candida albicans, differentiation was generally possible because they had different tonalities or halos with different colors. These pictures show tonalities so that you can differentiate them. Nonetheless, Candida tropicalis was difficult to identify in CHROMagar Candida Plus at 36 hours, because not all colonies were developed the metallic blue color described by the manufacturer. Instead, it had a pinkish color similar to Candida glabrata. At this time, Candida lusitania could also be mistaken with Candida glabrata, as you can see in the slide. However, at 48 hours, Candida tropicalis, Candida glabrata, and Candida lusitania show a different color, and we can identify them, as these photos show. At 48 hours, both media allowed an optimal identification of some Candida species. Moreover, CHROMagar Candida Plus allows the differentiation of Candida auris, glabrata, albicans, crasei tropicalis, and lusitania. In this plate, we can see the different colonies and species. We calculated sensitivity and specificity for all the 95 isolates, Table shows how the sensitivity and the specificity for both medium are the same in Candida albicans, Candida crasei, and Candida tropicalis. However, CHROMagar Candida Plus improves the sensitivity and the specificity in the detection of Candida glabrata. For Candida auris, Candida lusitani, and Candida parapsilosis, there is no data of sensitivity and specificity in CHROMagar Candida medium because this medium doesn't differentiate them. But I want to highlight the good results in the identification of these three species in the CHROMagar Candida Plus medium at 36 hours, and emphasize the importance of Candida auris detection, mainly in the context of an outbreak. Given the importance of detecting Candida auris in the prevention and control of an outbreak, we calculated the sensitivity and the specificity of both media only in positive surveillance samples for Candida auris. A total of 14 surveillance samples were confirmed to be positive for Candida auris. Although the number of samples are low, we obtained excellent results at an incubation time of 36 and 48 hours, as the table shows for CHROMagar Candida Plus. And in 24 hours, the specificity improves significantly in CHROMagar Candida Plus. By contrast, CHROMagar Candida had a low specificity for Candida auris, as expected, since this medium doesn't differentiate it from other species growing white and light pink and always have to identify by mammally. We are currently conducting a multicenter study in which 3 hospitals are already enrolled. We are evaluating the CHROMagar Candida Plus plates with a considerable number of surveillance samples and environmental samples. We hope to get good results soon and share them to the professional community. In conclusion, CHROMagar Candida Plus detects and differentiates Candida auris in addition to other major clinical Candida species such as Candida albicans, Candida tropicalis, Candida glabrata, and Candida crasei. CHROMagar Candida Plus medium could allow a reliable identification of Candida auris in only 36 hours, although it has got an acceptable sensitivity and specificity at 24 hours. Candida albicans and Candida crasei could be presumptively identified at an incubation of 24 hours. Candida tropicalis and Candida glabrata need 48 hours to achieve a good colorimetric identification. Last but not the least, identification by MALDI can be carried directly from a colony. No need of subculture. Thank you for your attention.
Okay, thank you very much, Carme, for sharing your experience with our CHROMagar Candida Plus. We are very glad that you find it useful also for screening purposes. Okay, now is the turn of England with the presentation of Professor Andy BORMAN from the Public Health England. By the way, Andy and his colleagues were the first to publish about the CHROMagar Candida Plus. His presentation, CHROMagar Candida Plus, a novel chromogenic agar that permits the rapid identification of Candida auris.
Hello, I'm Professor Andy BORMAN. First of all, I'd like to thank the organisers for giving me the opportunity to present some of our data today. Well, I'd like to show you our experience of CHROMagar Candida Plus, which we believe is an excellent novel chromogenic agar that permits the rapid identification of Candida auris. Now, most of the data I'm going to show you today has been published, but at the very end, there's a little bit of recent real-life data as well, which is novel. So for those of you who are not au fait with Candida auris, a little bit of background. It was initially identified and described as a novel species in 2009 from a single case of a patient with an ear infection, and this association with otitis was confirmed the same year in a series of 15 patients in South Korea. It's been subsequently reported worldwide from every possible clinical scenario, from mucosal superficial infections all the way through candidemia and deep seated candida infections. And it's been reported, as I said, worldwide and in hospitals across several continents, and in many of those countries it's caused large outbreaks. We know now it's got a propensity for patient-to-patient spread and causing these nosocomial outbreaks, and it's very adept at persisting in the hospital environment. We also know in certain countries and certain outbreaks, at least it's been associated with high crude mortality rates. Most, but not all, isolates are fluconazole resistant, and there are some multi-drug resistant strains, and we know that resistance actually is acquired or develops very quickly upon treatment. And this last point is also key. It's difficult to identify. It's a member of the quite uncommon Candida hemolonia complex, and it's frequently misidentified in commercial tests as a whole variety of other species, including hemolonia, but things that are more common and often things that are associated with superficial sites in humans. So as I mentioned, it's caused outbreaks worldwide. It's emerged globally and simultaneously on several continents. Here at the top, you've got the original description from Japan and Korea. Since then, shortly afterwards, it caused lots of multiple hard-to-control outbreaks reported in India and Pakistan with very high mortality rates. About a year later, reports from South Africa and Kenya, which is still ongoing to date, of nosocomial spread in huge outbreaks involving several thousand patients across the African continent, and slightly more recently parts of South America with Venezuela and Colombia. Again, multi-center outbreaks with associated high mortality rates. There have been also large outbreaks in many parts of Europe and the US, and this is predominantly or always with imported strains, and I'll explain what that means in the next slide or so. And there have also been a number of countries which have had sporadic introductions across most of the world. Russia probably should move now into large outbreaks because there were outbreaks in a big ITU in Moscow a couple of years ago. As I mentioned, this is simultaneously emerged on 3 continents. We know, well, this was the Sentinel work published by Sean Lockhart and his group at the CDC, where he looked at whole genome sequences of a variety of isolates from different countries. And to cut a long story short, it appears that this organism was simultaneously emerged on 3 continents as very, very geographically restricted clonal lineages. So all of the isolates from India, Pakistan were very, very similar, with unrelated isolates differing by less than 60 SNPs over a whole 20 megabase genome. Similarly in South Africa and in South America, but with tens, if not hundreds of thousands of SNPs differing between these isolates. So these have something that's been geographically constrained and fairly dormant on several continents for millennia, and then it's simultaneously emerged in the last 10 or 20 years. So, as I mentioned in the first slide, it's difficult to identify. It's a germ tube negative yeast. It produces only rudimentary or no pseudohyphae, although that's somewhat strain dependent. And as I said, it can't really be identified by conventional biochemical assays like API, Auxkiller, Vitek, yeast2, etc. This is what it looks like on conventional CHROMagar after 7 days. It's fairly innocuous. It's pale cream with a bit of a lilac center, but so are a lot of other Candida and related species. We know it can be identified by MALDI-TOF with the Vitek mass spec from bioMérieux once the databases have been updated with appropriate reference spectra. Otherwise, it misidentifies with this whole collection of organisms. And the Brucker MS, again, with existing database, but frequently with poor log scores. And then if you add in-house MSPs, then you can get these scores to an acceptable level. And obviously, rDNA sequencing of parts of the rDNA cassette has always been, for most Candida species, the gold standard. So how does it spread and persist? Well, we know that colonized patients often harbor the organism in the axilla and groin. This is from data presented at ASM Microbe a couple of years ago, where Sexton et al. Showed that from the armpits and groin swabs of 28 patients, there were variable, extremely, often very high numbers of CFUs per mil of auris in these hot, sweaty regions. And they showed that bioburden positively correlated with the amount of Candida auris those patients were shedding into the environment. Sarabi Traore and others, including myself, went a little bit further a couple of years later. This was the Candida auris outbreak in King's College Hospital in London, where they actually did settle plates where they measured the amount of Candida auris that was shedded into the environment, and they looked directly at what they called onward attack rate. So how many percentage, or what percentage of patients who were contacts of these were actually go on to acquire colonization. And to cut a long story short, there was a direct correlation between how much you colonized, how much you shed, and how much you pass on, which is not rocket science really. And this problem is, this is a Settles plate, or a typical plate that you would actually get from plating out an axilla or groin swab, and you recover lots and lots of Candida organisms of different species with different growth rates, different sizes. And one of the major problems is you would have to actually identify every single one of these colonies to formally exclude that the patient was colonized with Candida auris, which obviously is a huge problem and a very onerous task. So because of those difficulties in identifying every single colony, a variety of different labs have set up or developed different approaches to facilitate detection of auris in clinical samples. There are two basic different systems that we use. One was selective media, usually involving high salt and growth at 40 degrees or higher, because Candida auris is a halophile. The idea being it would suppress the growth of most or all other Candida species. And the other one is based on PTR and PCR assays, either real-time PCR or gel-based ones. They all work, they have good sensitivity and specificity, but the problem again is these are quite onerous. They require specialized technology and or some mycological expertise, which is where we think CHROMagar Candida Plus has the real advantage. So this next slide, this is actually a publication from a couple of years ago in Medical Mycology. I don't want to go through it in detail, but this is the appearance on Candida CHROMagar Plus of a variety of 52 comparator yeast species and also all 4 clades of Candida auris, all grown as single spots and incubated at 35 degrees for 36 hours. And to talk you briefly through the slides, spots 1 to 10 on this top left-hand panel are the 4 different clades of Candida auris, and then these are the top rows again on the next 4 panels are the same clades reproduced. And to cut a long story short, with one exception, which I'll come to in a second, all of these other Candida and related yeast species showed distinctly different morphologies than this quite distinctive auris, which is a pale creamy blue colony with a very distinctive and clearly visible blue halo. The only exception to this being spot 57, which is here, which is Candida dudensii, which is a vanishingly rare Candida species. At the Mycology Reference Lab in the UK, we've seen one example of this organism in the past 10-15 years, and we have identified tens of thousands of Candidas in that time. So it really, I don't think, is something that's too worrisome, but this is indistinguishable in terms of colony. So this is, as I said, single known organisms spotted on agar. What happens in a real life situation or a pretend real life? Well, this next slide was basically taking known organisms, mixing them together in different proportions in saline and swabbing those bits of saline in parallel onto Candida CHROMagar Plus on the left-hand side of each panel and Sabourates agar on the right-hand side, and incubated at the temperature 35 degrees as described by the manufacturers and for different amounts of time. And there are several take-home messages here. One, of course, is in all combinations. This is auris and cerevisiae. The auris is clearly distinctive and differentiable from the other organisms, so auris and the blue halos. What's more important is actually when you look at the equivalent plating out on Sabourates after 36 hours, you have a few colonies only, and these are all correspond to Saccharomyces cerevisiae. And if you look at the bottom-hand panel on the left-hand side, these large, huge colonies now after 60 hours are cerevisiae, and it's only after 60 hours that these smaller colonies of auris are becoming apparent. And what you will also see just by eye is there's a lot more auris recovered on the CHROMagar Plus than on the equivalent Candida plates, Sabourates plates. And the same is true for all combinations we looked at. On the right-hand side, the top, auris and its close relative Humuleniae. This is after 36 hours and 60 hours, where auris is only becoming apparent after 60 hours. And again, it's clearly distinctive on the CHROMagar. The next panel is auris and Trichosporin inkin. Again, much more auris on the CHROMagar than the tiny colonies on the Sabourates. And the bottom one is auris and Paracellosis, another organism commonly found as a skin commensal. And again, much more auris recovered on the CHROMagar than the Paracellosis, despite the incubation period on the Sabourates, being almost double. So we're fairly sure that actually, in all experiments, that both the number of colonies and the speed of growth of Candida auris was substantially increased on the CHROMagar compared to conventional, in our case, Sabourates agar. And in some cases, that increase was up to twofold, so 200% increase in colonies, which is non-negligible. We have recently had the opportunity to have a dabble at real-world experience with CHROMagar Candida Plus. This was a very recent case of March 2021. This was a UK patient that returned from Egypt where they had been hospitalised. They presented to the emergency department directly from the airport when they arrived in the UK because they were acutely unwell. And on admission, they were found to be colonised with MRSA. They had VRE, They were COVID-19 positive, and they were also colonized with Candida auris, and they went on actually to develop candidemia following a line infection with Candida auris. Because of this nasty cocktail, they were actually barrier nursed into the side room on arrival in a large 24-bed ward with incredibly intensive infection control protocols. They weren't actually particularly ill with the COVID-19, so this probably reduced the likelihood of transmission and onward spread, because the nursing staff were able to adopt normal infection control protocols rather than the ones that we've seen all of the news of wearing the same PPE for 24 hours at a time. Nevertheless, they were worried obviously about transmission and also the potential for environmental contamination. So they sent swabs to us from all the patients on the ward and various other environmental samples, and this is what we got. So a variety of scenarios. We had a culture negative patient, axilla and groin swab. The bottom is 4 different patients, all colonized with different quantities of Candida auris. You can see just how much yeast some patients on ITU are colonized with. Then the top row, we have a patient colonized with Nicasiamyces ex Candida glabrata, and on the right-hand side, this is a mixture of Candida parapsilosis and Lusitanii. And then the one panel of interest to us was an environmental swab. It was the only place in the whole ward where we did recover auris. None of the patients were positive apart from the index case. And this was 2 colonies of auris actually recovered from the chair in the index case patient's room. So that was quite reassuring that there wasn't any onward spread. And this really just showed the power of CHROMagar Plus to actually be able to spot this organism. So with that in mind, I'd like to finish by, well, the conclusion's fairly obvious. We believe that the distinctive colonial appearance is fairly unique to Candida auris on Candida Plus agar, and we've shown that the rate of growth is enhanced and colony yield is increased compared to conventional mycological media, which we think actually makes CHROMagar Candida Plus an ideal primary isolation media for the detection of auris. And it just remains to thank CHROMagar for actually supplying the media that we use for these studies, Sean Lockhart, CDC, and Ronan Ben-Ami Tel Aviv for gifts of the isolates of the South American Israeli clades, and the other members of the MRL in Bristol, Prof. Liz Johnson, our director, and Dr. Mark Fraser, who helped me with these studies. Thank you very much, and I'll be happy to take questions at the end.
Okay. Andy, thank you very much for sharing with us this very interesting information about Candida auris. If I'm not mistaken, you are now studying the relation between COVID-19 and Candida auris. Maybe when you finish this work, we will be able to do another webinar so for you to present your findings.
Absolutely, yes.
Okay, so this is for the presentations of today. Now we have a few minutes for a few questions. And as I said, if we do not manage to answer to all the questions today, you will receive a reply via email. Okay, so one first question, I believe this is for Carme. What detection method was used as the gold standard in the determination of CHROMagar Plus sensitivity and specificity for Candida auris detection? This is from Nasiru Abdulhai.
So, for the study, we use CHROMagar Candida Plus, and we use also CHROMagar Candida. We test and confirm all colonies with Malditov, Rucker, in this case. And we calculated the sensitivity and the specificity in order to the results of MALDI. Moreover, we usually in our workflow in the laboratory, we test the resistance to fluconazole by VTEC in the case of surveillance samples, and with sensitivity in the case of clinical samples. So the reference method to confirm the colonies was mass spectrometry with MALDI.
Okay, thank you very much. In fact, this question about the confirmatory test came several times. Yes. There have been also several questions about where to get the CHROMagar Candida Plus. So you can go to the contact page in our website to find the local distributor. If you don't find it, write to us and we will address your inquiry to a local distributor. There were also some questions about the publications that were presented during the webinar. This also can be found in our website. Okay, a question, I believe this is for Nuria. Thanks for the presentation. If a researcher wants to screen patients on admission in wards, what are the sampling sites? Can urine be used as well? Are immunocompromised patients, diabetic or HIV positive, more susceptible? Well, there were a few questions in one question.
Thanks for the question. Normally, the sites are growing, axilla. We also use pharyngeal samples because we also look for gram-negative multi-drug resistant microorganisms, but it's axilla and groin, maybe nasal samples, but I think it's more in axilla and groin. In our case, as we have the outbreak in the intensive care unit, normally we perform all the axilla and rectal and pharyngeal samples to all the patients at admission and once weekly to all the patients in the ward. And then sometimes when we have patients that are readmitted or previously colonized or coming from other institutions, we have the possibility to perform a direct PCR. We have 1 hour PCR and then we perform it from the same swab. So that's what we normally do. And for HIV, well, immunosuppressed patients, we don't really have a relation of Candida auris colonization with HIV patients because it's just the patients that go to the ICU. So we don't have a high incidence in especially HIV patients. Maybe diabetes, there are more patients, but it's not, I don't believe it's a cause of immunosuppression. It's more abdominal surgery, patients who are with a lot of antibiotic treatment, with a long period of intensive care admission than being immunosuppressed. At the end, they get colonized, and then if they are a lot of time in the ICU, they have candidemia or other infections with more catheters or a urinary catheter. It's more this kind of patients, at least in our setting.
Okay. A question, I believe, for Andy. Is there any evidence of difference of virulence between the clades?
Yes, there is. We've published a couple of papers, the first using Galeria, so the wax moth larvae, which showed that the Indian and South American clades were much more virulent than the South African and Japanese Korean. And that was recently confirmed in a collaboration between ourselves and a group that we work with extensively in Hungary, where they actually used a neutropenic mouse model and found the same thing, that definitely the Indian and South American clades seem to be more virulent.
Okay.
Can I just add as well to Nuria's answer, which I agree totally. I mean, from all of the from all of the UK outbreaks from a few years ago, we found definitely that the axilla and the groin were the best sites by far. We did see patients who were colonized in the urinary tract, about 10% of them, but if you want to capture the maximum, it's axilla and groin, and we would agree as well, it's important obviously to screen them on entry, but often patients who are colonized when they first appear in the hospital, they're not actually expressing enough the Candida auris to be able to detect it. So screening again after 1 or 2 weeks on IT when they've had lots of antibiotics is the best way of capturing all of the patients who are importing it into hospital. But yeah, totally agree with our answers.
Okay, a couple of more questions from Mohammed Elmi Aziz. We found colonies similar like Candida auris with blue halo around its colony. However, when we performed Vitek 2 3 times, the results were Candida emuloni or Candida emuloni variant vulnera. Okay, I can give just the beginning of a reply. This is microbiology. Despite the medium is super specific, you can always get a microorganism that can have this kind of behavior. But as we discussed, there have been also reported misidentifications with the biochemical tests. So maybe the advice is to do a sequencing if you want to really make sure. Maybe Andy, you want to add something?
Yeah, no, yes, please. I mean, yeah, they are similar. We did do Hemoloniae and the other members of the Hemoloniae complex, and they are similar. You can distinguish them. We think auris gives a much more distinctive halo than Hemoloniae, but in real terms, Hemoloniae colonization is so vanishingly rare in clinical samples that if you're forced to mold it off to distinguish Hemoloniae and auris, it's still a major bonus to have Candida Plus, CHROMagar. You're getting rid of everything else. I mean, I think we've seen throughout all of the outbreaks of auris in the UK, we've probably seen 2 patients where they were actually colonized with Hemoloniae rather than auris. So that's a vanishingly low numbers. So I don't think it's a massive issue in a clinical setting. But we thought we could distinguish them with a bit of an eye of faith and holding them up to the light and a following wind. It's easy to, if that's the only things that cross-react, it's easy to identify by moldy stuff, what you said.
Well, for us at the beginning,
okay.
when we used VTech, we had Candida emulonic results, and then with the mass spectrometry, it was Candida auris. And it was after that confirmed it was Candida auris. But VTech had wrong results.
It's okay for resistance, but
not for identification.
Okay. Yeah, as microbiologists, always double check. Okay, I think we have, okay, one more question. Susan Sharp. Oh, Susan from the US. Is it necessary or preferred to use liquid pre-enrichment culture prior to plating to CHROMagar Candida Plus? Nuria or Carmen, maybe with your experience?
We try the liquid medium, and we don't show any difference between directly culture or in a incubation previous in liquid medium.
Okay.
In our case, we don't have any. We haven't any difference.
Okay, so yeah, maybe no interest in pre-enrichment. Okay, I think one last, Andy, I believe this is for you. You have highlighted the superior recovery of the CHROMagar Candida Plus when compared to Saburo for Candida auris, 150%, 200%. Is this true also for other Candida species?
We haven't tested every Candida species, but for the ones that we made those mixtures, so Hemoloniae and Cerevisia, it seemed to be that the Candida CHROMagar Plus was actually suppressing a lot of the more common Candida species and preferentially growing the auris, which I think would agree with what Nuria was just saying, that there probably isn't a need for a pre-selection stage because actually Candida auris seems to really like this agar. Which is why we would propose it as a primary isolation media rather than Saprodes. It grows faster, the colonies are bigger, you get more colonies, and that seems to be the opposite with a lot of other Candida species. So I think it looks almost suppressive for non-auris, at least the ones we looked at, which is great.
Okay, very interesting. Thank you very much again to our 3 speakers. Again, not only for the presentation of today, but for having being so courageous to evaluate this tool during the peak of the pandemic. This is all for today. Oh, I just have been informed by the control tower that there were 46 countries represented today by the attendees. So thank you very much again, if you have any more questions, don't hesitate to write to us at chromagar@chromagar.com. And okay, please keep safe.
Thank you. Thank you, Alberto.
Thank you.
Thank you, everyone.
Bye.
Bye-bye.
Bye.